All right, everybody. Today we are going to talk about gene regulation or the control of eukaryotic genes. So it's just like important to remember that today we are talking about eukaryotic cells and not prokaryotic cells. Remember that prokaryotic cells use an operon system. And today we're talking about how eukaryotes control their gene expression. And so kind of just the big questions that we're thinking about today is just the main one: how are genes turned on and off in eukaryotes? So when, how do we manage when different proteins are going to be expressed or not? And then finally, just how this process can allow for differentiation between cells and how different cells form specialized functions. So it's just kind of important to always be thinking in the back of your mind that, a neuron does not perform the same function as an epithelial like skin cell. And so those cells are going to be activating or expressing different proteins. And this is kind of the process or why cells are able to form these specialized functions because they can turn their genes on and off differently. So the evolution of this process it kind of happened for different reasons. So prokaryotes, they have evolved to grow and divide quickly. They have to always be changing to the types of environments that they're in, and they have to be able to quickly adapt to those environments. And so their system, while it might be a little more simple than a eukaryotic system, it is able to make those organisms function efficiently and respond rapidly to their environments. Gene regulations, their gene regulation really is about like turning their genes on and off based on kind of what they need at the time. And so it's just important to kind of think of like that lac operon, how bacterial cells are able to, turn on the expression of the lactase enzyme when they have to digest lactose. And so, that's kind of like the need for that type of gene regulation. So eukaryotic cells have kind of have evolved this gene regulation system for many different reasons in their cells. So one reason is so that their bodies can maintain those internal conditions, that idea of homeostasis. Think about like temperature control, think about different body systems controls. So when we face differences in external conditions, our bodies can maintain kind of that happy place. And then also so that they can regulate the body as a whole, so that cells can grow and develop, so that cells can specialize and have differences based on the type of cell that they are, and that those cells can perform the functions that they're meant to do. And so that's really how the evolution of gene regulation has been different in eukaryotes and prokaryotes. All right, so we're going to talk kind of about the different levels of regulation that can happen in our cells. And so we're going to start at like DNA control, then we're going to talk about transcriptional control, and then we'll talk about translational control. And so at any step in the process, our genes or our expression of proteins can be controlled. The first type of control we're going to talk about is chromosomal regulation. And so this happens in the nucleus because we are directly kind of regulating the DNA based on whether it's open or not. And so why this happens is it's kind of that first line of regulation. Like, is the DNA even going to be able to be transcribed? And so in some cells, they'll have regions of their DNA where they don't need any of those proteins to be transcribed because they're not using those proteins. And so that DNA might be packed more densely than areas of the DNA that has genes that needs to be transcribed. And so that will be packed more loosely. And so this degree of packing is what regulates whether or not the DNA is transcribed or not. And so remember that our DNA wraps around these proteins called histones. And so when the DNA is tightly packed and coiled around those histones, it's not going to be able to be transcribed. And so when the DNA is unwrapped around those histones, it's able to go through that process of transcription. So there's kind of like two different ways that DNA can be regulated at this level. And the first is something we call DNA methylation. And so when DNA is methylated, this means that there is not going to be any transcription of those genes. The genes are essentially turned off. And so what happens is methyl groups or CH3 groups bind to cytosine. And so what that does is it inactivates the genes around those areas of DNA that are methylated. That DNA is going to be tightly packed around those histone proteins. And so they're not going to be able to be opened up and transcribed by transcription they won't be able to have transcription factors bind, and then promoters won't be able to bind, and RNA polymerase won't be able to copy genes there. And so you can kind of think of this as like the inactivated X chromosome, the bar body. It's just like really condensed areas of DNA that are not going to be transcribed. Now, kind of the opposite of that is acetylation of histones. And so in acetylation of histones, that's going to actually unwind the DNA. It's going to make that DNA uncoil and be loosely wrapped around those histone proteins. And so that's actually going to enable transcription, and those genes will be turned on. And the way this happens is just like the methyl groups made the DNA tightly pack around the histones, acetyl groups are going to attach to the histones, and that changes the conformation of those histone proteins so that they loosen up and don't wrap the DNA around them as tightly. And so when the DNA is more open, then transcription factors can bind. Transcription factors can pull together that promoter complex, and then RNA polymerase is able to transcribe those genes. So the field of epigenetics is really interesting and is something that's kind of like blowing up in the scientific community. And it's this idea of kind of studying the changes of gene expression based on these tags. So these methyl groups and these acetyl groups that get put onto the DNA and the histone proteins, those can be tracked over time. And so what scientists are finding is that these tags, or these methyl groups and acetyl groups, can actually be passed on from one generation to another. And they think that this methylation and acetylation is actually environmentally controlled. And so these groups can be added based on things that are happening in an organism's environment that might, cause them to have more methyl groups or less methyl groups, or more acetyl groups or less acetyl groups added to their DNA. So there's this really interesting study with the descendants of Holocaust survivors and having kind of like altered stress hormones, where they've actually turned off those genes. They've added more methylated regions around those stress hormones so that was not expressed in their DNA, and they've actually passed that on to their ancestors. So that's something that we can track in the descendants of Holocaust today. And interestingly enough, their descendants actually have a harder time coping and have more like stress disorders because they don't activate those stress hormones. And so they have more issues with that today. And so there's this, I will put a link to this, to this in Canvas if you would like to go and kind of read up on that later. It's a really interesting article. And then this TED Ed video is just a really good video about what epigenetics is. So if you're interested, you can access this PowerPoint through Canvas and go and take a look at that video. The second level of control we're going to talk about is transcriptional regulation. And so this happens again in the nucleus because that's where transcription is occurring. And kind of what's happening here is this is how genes are turned on and off based on chemical signals. And so based on, if you recall when we were talking about in Unit 4, cellular communication. And so remember that cellular communication can kind of activate some of these transcription factors or activators of transcription when, cells have communicated that a certain protein needs to be made. And so this just controls gene transcription, so the transcription of certain genes. So kind of our example here is just the activation of that complex that will start transcription. So it all starts with a transcription factor. And so remember, those are those proteins that can be signaled through chemical signaling to go into the nucleus to bind to DNA. And so when a transcription factor binds, that turns this gene on. It means, it's going to be made. And so that transcription factor is going to bind to that promoter region, which is upstream of the gene. And then other proteins can also kind of be worked into this equation as well. And so this is where you'll see enhancer regions becoming important. So an enhancer sequence is kind of a distant control, and activators are another type of protein that can bind this enhancer sequence to help activate or turn a gene on. And so as you can see kind of in this picture, you have your activator, you have your transcription factor. They're forming kind of this bonding complex with the DNA that's going to recruit RNA polymerase over, and then RNA polymerase is going to start to make that messenger RNA. Silencer proteins, this is just another type of protein. So just like we can activate this process, we can also turn it off. And so if silencer proteins are recruited into the nucleus, then that's going to, turn those specific genes off so that they won't be expressed. Okay, the third level of regulation that we can have is something we call post-transcriptional control. And so if you remember, we talked about that, like after mRNA is made, there are some modifications that have to happen to it in the nucleus before it can be translated. And so one of the ways that we have mRNA changing is by removal of the introns. So if you see in this first picture here, we have a bunch of introns, those are being removed, and then all the exons are coming together, and then we have that 5' cap and that poly A tail to kind of protect that mRNA transcript. So in post-transcriptional control, again, this is happening in the nucleus. We haven't left the nucleus yet, but this is where this kind of this RNA splicing can take on different forms. And so what that means is just the RNA gets spliced together in different ways. And so we're going to have variable processing of exons, meaning the exons might not always come together the exact same way every single time. So if you look at this picture over here, you can see, there's one, here's like some exons in this DNA. So you have exon 1, 2, 3, 4, and 5. And so after RNA splicing, when we remove these introns, sometimes exons can be removed as well. So if you're kind of looking in this first transcript, right, we are putting together all these exons, except we're also removing exon 4 to create this final RNA transcript. In this kind of second example, we're removing exon 3 and then putting all these other ones together to make a different RNA transcript. And so this is just kind of combining the exons in different ways and like omitting some, one or more of them to kind of give even more versions of genes. And so that's just kind of another way that genes can be changed from what maybe the original DNA sequence says. Okay, so now we're moving on to translational control. And so in translational control, I'm sorry. In translational control, this is happening in the cytoplasm. And so we finally have this mRNA transcript. It's made it into the cytoplasm. It's ready to go to a ribosome to be translated into a protein. And so this is kind of how cells decide how long is mRNA going to stay around. Like, what's the lifespan of this mRNA molecule going to be? So messenger RNA doesn't hang around forever. It'll be around as long as it's needed to make, an adequate amount of protein. And then once it's not needed anymore, it's going to be silenced or cut up. And so the way that this happens is by something called RNA interference, or siRNA. And so these are just short segments of RNA. They're only like 21 to 28 bases long. And what they do is they bind to mRNA and they kind of act as this death tag. And so it triggers the degradation of that mRNA molecule, or it tags it for death. And so what it does is it effectively silences that gene and makes it so that that protein isn't going to be made from that mRNA segment anymore. This video down at the bottom right is just kind of like another TED Ed video that just does a really good job of explaining kind of how these siRNAs work and kind of what their level of control is in the cell. And then the last way that we do regulation of genes is by something called post-translational regulation. And so this is kind of two ways that proteins can actually be regulated inside the cell. So after a protein is made, well, first this is occurring in the cytoplasm because that's where proteins are made. And so proteins can be made and then they are processed. So we know that after a protein is made, it has to be folded. So there's all those different levels of kind of protein organization. So this would kind of be that tertiary and quaternary folding to process that protein so it's ready for its final job. There might be sugar groups added to it. It might be targeted for transport to a certain area of the cell or outside of the cell. So that's kind of one way that proteins can be altered after they're made so that they can go on to do their job. Now, kind of the opposite of that would be protein degradation. And so if a protein's not needed anymore, if something's wrong with it, then it can kind of be tagged for death as well. And so the way that proteins get tagged for death is by something we call ubiquitination. And so ubiquitin is just a tag that can be put on proteins. And then those tagged proteins go into something called a proteasome. And those proteasomes actually just take the protein apart, like cut it into different amino acids. And then those amino acids can be used to create, another protein again. But that's kind of a way that, if we don't need that protein anymore, that this process can be regulated. So that's all I have for you today in terms of gene regulation. If you have any questions, please.